Okay, thanks for the introduction, and thanks a lot for the studium to give us the opportunity to present our ongoing work on nasal vaccination against COVID-19. So I'm here to represent all the team of Professor Isabelle Dimier in Tours. And this project, in fact, started a couple of years ago now, when there was the beginning of the pandemic. And with Isabelle, we were working from home and discussing about how frustrating it was to be locked at home and thinking about the vaccination field, because we were really into it. And there was this project going on with vaccination in zoo of different kind of species, using a strategy by intranasal way, with a nice carrier and a nice antigen. It was against toxoplasmosis. It's still going on in different zoos in France and even out boundaries. And we said, okay, we're a small lab here in the middle of nowhere, Tours, you know, a nice place. But okay, why not try to do something? And there was this call from the French agency, ANRS, and we said, why not say, okay, because we've got this working in a species very close to non-human primates, maybe it could be very interesting to try if it might help to protect against COVID. But also, at this time, we were really focused on, okay, we want to go back to normal life. So we want to block contagiousness, okay? Not only protect. Obviously, it's the first important step, but if we can block contagiousness. So, is it working? Is it on? Okay, should work better now. Okay, so I won't go into detail with all those very frustrating and boring numbers here, but you're all aware of the pandemic and the fact that there is a lot, like almost every day, we hear about a new variant, okay, going on. Mostly more contagious and hopefully less, with less impact in terms of pathology, but still very contagious, okay? And what's really interesting is to think that right now, okay, we have, I think, in Europe, 5 vaccines that are allowed on the market that are finally pretty efficient. Even if we check here the low efficacy against the new variant, they still kind of protect with the repeated boost against the more drastic pathology. But the point is, they don't last very long in terms of efficacy, and they don't block contagiousness. You've all heard people that were already vaccinated that were infected later, or people that were vaccinated but that were capable of transmitting the disease. So that's a real concern, and wearing those masks, okay, every day is very frustrating. And the idea also is a way that they're all based on solely one protein, which is suggest to a variation and very critical. And these were all features that were very, very sensitive in terms of vaccination. So how can we stop the pandemic? So in our lab, we were working for a long time now on the goal of achieving a nice mucosal vaccination. What is the goal, in fact, is if you consider classical parenteral vaccination, you induce systemic immunity. So mostly within the blood, you can really figure out the level of antibodies there, but you lack the high immunity at the site of entrance of the virus, okay? But with nasal vaccination, you can induce at the main entrance of the virus a strong blocking immune response, mostly with the IgGA, which are important antibody here capable of really blocking the entry of the virus. And it's, I think, an interesting goal. And the idea also was to try to be more useful against several strains of coronavirus. So here it is. So the goal was, in fact, to have the appropriate immunity. So we all hear about neutralization, humoral immunity, antibodies, but also the cellular arm of the immune system is very important. I won't go into detail today because I should confess first that I won't show you any nice table, any nice data, because everything is ongoing under a patent and it's not published yet. So I will just explain to you what we did, and you have to trust me on the results that we have, okay? And the point is the route of administration. So the goal is to induce both mucosal immunity at the entrance of the virus, but also systemic immunity, obviously. The antigen. So the idea was to have something not only based on the spike protein, but something that induce antigens that won't change every time, okay? And a good vector, because now more and more we hear in the news about, okay, the instance, the World Health Organization say, we need intranasal vaccine now, we need this immunity, mucosal immunity. But okay, scientists working on the field, they thought about it a long time ago, okay? The point is, it was very tricky to find the appropriate vector, something that help the vaccine to be efficient in the nose. Because obviously the nose is the main entrance for pathogen. So in fact, it's not a nice area for a vaccine to stay and induce an immune response. It's obviously very tricky. There is a short window to be efficient, and there is bypass to really be able to do so. So this is mostly the formula of the vaccine. The idea is to use what we engineered here in our lab. So the spike protein is a very good antigen, so we keep it, obviously. But we had some protein that won't be subject to a mutation, okay? And we mix this fusion protein with what we call mucoadhesive nanoparticles. This is kind of the trick here. The idea is based on maltodextrin. In fact, it's mashed potatoes, okay? And the point is, it associates with the antigen. So the antigen is kind of melt with those nanoparticles. It's not really within the nanoparticle. It's clearly embedded with those nanoparticles. And it allows the adhesion to the mucosa, even with the ciliary mechanism, physical and also chemical, that is in the nose that block the entrance. It allows the vaccine to be sticky within the nose in the appropriate area of the mucosa where the immune system is. And obviously what we're working on now, that this is the third part of the vaccine, it's the appropriate device to deliver the vaccine, because obviously it's not a needle and a syringe here. It's a system of spray that will allow a shot of the vaccine within the mucosa. And obviously, it was very tricky to use it on rodent, on animal models, so we bypassed this mechanism in the preclinical studies that I'm going to talk about, but it's something that we're really focused on right now to go for the clinical trial. So the experiments that we perform to validate the efficacy of the vaccine are mostly of three types. The first were in classical laboratory mice, just to check the induction of an immune response, because the classical standard lab mice is not sensitive to the SARS-CoV-2, okay? But she can mount an immune response against any antigens if it's delivered properly. So the idea was to level the antibodies response, the cellular response, within the systemic and also within the mucosa. That was very important to us, within the nose and the bronchoalveolar lavage. The second step was to check a laboratory freak, the K18, which is a laboratory mice designed to express the human receptor for SARS-CoV, but mostly broadly within the body. So in fact, it's even more sensitive than human to the virus, because it expresses really broadly the receptor. So it's a very stringent model. If we manage to make those mice survive with our vaccination, it's very, very promising. And the last and most critical model is the Syrian hamster, because it works very similar to the human in terms of pathology. It expresses the same receptor in a similar manner. It expresses the same pathology pattern and duration, and it's a good way also to check contagiousness, because mice, they don't spread the virus by the nose, okay? They don't cause. The hamster do, okay? And you can check contagiousness between animals with the hamster model. So the immunogenicity, to go briefly, we were able to check after the same pattern. That's important. We do 2 vaccinations 3 weeks apart, 2 doses, okay? In fact, in a similar pattern to what has been done to us at the beginning of the global vaccination. And then 3 weeks later, we check even the immune response or the protection, depending on the model. So for immunogenicity, we were able to measure the amount of antibody in the blood, in the nasal mucosa, and also in the lung. And it reflected a good and also efficient, because we did neutralization assay against several variants, okay? The spike protein that we have in our vaccine is the original Wuhan one, okay? The first one. But even so, due to the addition of other protein and due to the way of inoculation, we were able to get robust neutralizing response within the whole body, but also a nice cellular immune response. And it's probably something that right now it's even for us difficult to decipher the importance of each part of the immune response, because it's probably complementary. And also we don't know what might work better in human, okay? We're still in mice. We have to stay humble about that. And then, because we were pretty happy with the result, we break our guinea pig money saver, okay, to buy some expensive K18 mice and perform experiment with challenge this time, okay? We perform the vaccination protocol, 2 vaccinations 3 weeks apart, and then we perform the challenge with the Wuhan or the Delta variant, which is more contagious and also more nasty, this one. And we were really happy to see that the mice that has been vaccinated, they didn't die of the challenge, but also they didn't show any clinical sign as compared to the placebo groups. So it was very encouraging. And then later we performed the same kind of challenge experiment with the hamster model, because it's also a good way to decipher the mechanism of the pathology within the lung mucosa and also within the lung. And also, what was really interesting here is we made 2 groups of hamster, 1 placebo, 1 vaccinated. We challenged everyone, and then 1 day after, when we know that the animals that are not protected are very, very contagious, we put a naive hamster to cohouse in the same cage, okay? In fact, we put 2 sentinel hamster per challenge hamster, and we check a couple of days later if the hamster, naive at the beginning, pick up the bug and get infected. And in fact, we had very, very nice result, because only the guys that are with a placebo challenge hamster get the disease. But the ones that are close and in the same cages as the vaccinated and challenge hamster, they're not infected. We don't find any trace of the virus within the lung, and almost nothing in the nose. So this is really interesting to us in a way that there is a protection, probably against a broad of variant of the virus, but also it can stop the contagiousness, sorry, of the virus. And I think it's pretty important. So after having fun performing science, now we're getting into the more boring part of the project. Exciting, because we're trying to put the project the more and more, but now it's about money, it's about politics, it's about regulation, but we're really deep into that right now. So what we managed to do is we had to make a spin-off, a startup from the laboratory to be allowed to raise money for the clinical trial. And right now we're working with a company, a French company, which is near Toulouse, GTP Bioways, and all the money that we raised from our first, from the beginning of the company, of the startup, was used to pay this company, which is developing right now the GMP formulation. It means the vaccine in a good manufacturer practice that is necessary to be allowed to do first toxicity on the rabbit model, to be allowed to go to the clinical trial after with the same batch of GMP produced vaccine. So it's very costly, it's not in our hand right now, but we have really to keep an eye every day on what they're doing, what is the advancement, because the calendar is very short to be on time, and it's very difficult, but it's quite interesting. So this is not my part, because I've got a colleague that is really specialized in the development of the protein and complex protein and is able to speak the same language, okay? I'm kind of lost in this part, but it's going pretty well right now because they managed to get the first clone that produce the protein properly, nicely, and now we have to see if the level of production is enough to go in an industrial way to get the GMP batch. And in the meanwhile, so we are planning the clinical trial. So everything is almost set up now. We just have to kind of to wait for the batch to be ready, and also the device. I'm going to talk about it later, the spray system to deliver in the nose. Because we know that the hospital from Tours and the hospital from Cochin in Paris will be co-leader of the clinical trial. We have almost set it up everything, so now it's only a question of paperwork, very complex to be done. But we've got a good scientific committee to follow the clinical trial. So we're kind of in the starting block right now, and we're developing the last part to be able to perform at the beginning, early next year, the clinical trial. Phase 1 and 2 combined if everything goes well. So I just mentioned it before, the device to deliver the spray in the nose. Also experiments that were performed in rodent were using a micropipettes, classical laboratory micropipettes, to deliver a small volume, about 10-20 microliter in mice, twice or 3 times more in hamster, which are the standard for nasal vaccination with those models. And for human, we need really to use something that might deliver a nice spray of the single dose that is used for vaccine, because there is a lot of company working on therapeutics by nasal way. But in fact, there is only one nasal vaccine on the market in Europe and in the US, which is the same against flu, okay? There is only one device in the market that is more adapted, in fact, for pediatric than adult, just to know. And right now, we're working with 2 companies, Aptar, which is a huge American company, and there is one part of the company working in France, and also Resica, it's a company that is part of the Resifarm system. Resifarm, there is a site next to Tours in Mons, which are producing the Moderna batch of vaccine for Europe. And the guy that was in charge of this company near Tours is now the CEO of our startup right now, because he was really convinced that our project was interesting. And he allowed us to be in touch with people from one spin-off of his company working on delivery system. And I'm in charge of developing the system with those people, because the idea is to use a modelization of the nasal cavity to really adjust the deliverance of the vaccine to the appropriate area where are the immune cells that we want to activate, okay? Because it's not like a therapy where you've got your own spray that you use every day for months to cure something. Here, you've got one shot per nostril and it's done. And you don't want to miss the shot, okay? So that's what's going on right now. And these are all the people involved just in the spin-off that we made to raise the money and to really follow the vaccine to the clinical trial. But obviously, it missed like 80% of the people that help working in the lab on the project. So this is Patrick Barrios, so the CEO that was working previously in Recipharm, Isabelle, the professor in charge of our lab, Nicolas Aubray, which is our specialist for the design of antibody and complex protein, and the newbie, Julien Desroyer, which is used to work a lot in pharmaceuticals, and is here the first new guy from Lovaltech to help us in the development of the process for industrial, obviously, if it works, vaccination. And just to show you where we are now. So, in fact, I discussed about the production of the GMP batch. So it's almost ready. So right now, what's very, very critical is to be able to be on time for the phase 1 and 2 clinical trial early next year. So what I need to mention now is, okay, it's more like advertisement, but it's what we use as basis to develop the virus. It's non-invasive, obviously, because it's based on protein, okay? What's interesting is, I didn't mention it, is that our protein and its association with the carrier is very stable, even at room temperature for several weeks. But you can keep the formulation efficient in the fridge at really a decent logistic temperature for years, okay, as compared to the RNA vaccine, for instance, that really need to a huge complex logistic, okay? So it might be used and designed as a booster for vaccination, because now there is already some vaccine on the market. And the idea is, I don't know if you're aware of that, but finally right now the people that are really well protected are the ones that had different kind of vaccination, or that were infected at one point and then vaccinated. Because if you multiply the way that you're interacting with the pathogen or with something mimicking the pathogen, it really boosts what has been started in terms of immunity. So also, because it's efficient per se to protect against the disease, it might be used in primary instance in countries that didn't get access yet to vaccination, because you know that in Europe and US, so, it's okay. But in developing countries, there is tons of people that don't get access to the vaccine. And because it's very easy for logistic, convenient for administration, and it might be really an interesting alternative for vaccination. And just to end, I would like to thank all the people that supported us. So, in fact, most of the funding that we had so far are public funding, and also because we were awarded of the deep tech by the BP early this month. I think we were also we managed to get money that will help us funding the toxicity test that will be performed on rabbit before clinical trial, and also the different institutes that collaborated with us on the project. And so this is just the starting of the advisory board that we have set it up for the Lovaltech company so far, with people that are really specialists in their field in terms of vaccination in France. And this is almost, we need to do a new picture soon with Julien that arrived in the team very soon. So thanks a lot for your attention, and I'll be glad to answer your question.
Let's address